So thank you guys for coming to the session. It's great to see such a packed audience, and I'm looking over the program. I'm really excited for everything that's going to go on at WMIC. So I'm honored to be a speaker. So I'm not going to talk about hallmarks of cancer or generic markers. I'm going to talk more about surface imaging cell surface receptors. So imaging cell surface receptors, or why I've been focusing on it in my career and why other people do, is for a couple of reasons. One, I'm an engineer, so I'm incredibly pragmatic. I don't have to get inside the cell. I don't have to pass a plasma membrane of a cell. I don't have to get into the nucleus. So imaging the cell surface is really great. But then also from a biology perspective, so I am a biologist, the cell surface really sits at that interface between what's going on outside of the cell and what's going on the inside of the cell. So if you want to know what's going on the outside, you talk to the receptors, and if you want to know what's going on in the inside, you talk to the cell surface receptors. And so I really feel there's a lot of information that can be gathered and garnered from understanding what's going on in the cell surface. And so I want to I know we're all here because we all buy into the WMIC, or molecular imaging concept, so I'm sure you guys are all the choir, and I don't have to really preach to you guys. But molecular imaging can be just more than early detection. We know we can study pathways, we can detect disease, patient treatment stratification, monitoring therapeutic response, drug distribution assessment, and monitoring recurrence. So this is our value proposition as molecular imagers. This is what we can really do and how we can fit into medicine and how we can fit into the pharmaceutical companies and entrepreneurship. So that's another thing that I really am here, here's my little soapbox thing, but getting the business community, getting entrepreneurial, most of us want to be translational. Molecular imaging lends itself to being very translational. And so being able to get these things into the clinic and to be used for patient treatment would be really great. So again, this is what I think our value proposition is. And I also want to say that as a biologist and as a target identifier, I'm agnostic to the modality. So this really speaks to the themes you'll hear throughout all of WMIC presentations, is that it takes a team, it takes a village to really make molecular imaging work. And so I'm more on the biology side. Like I said, I make the imaging agents. But there's technology, there's all sorts of great clinical applications that have to work. And so for me, I'm more agnostic to what this is, but that doesn't mean I can be blind to it. I can't choose pairs that are the wrong type of pairs. And so really working with people that are the physicists, if you're not the physicist, or the biologists, if you are the physicist, to come together and make these imaging agents that can be clinically approved is really going to be valuable. So in order to do molecular imaging, you have to think about two things: endogenous versus exogenous probes. We're really not looking at anatomy anymore. We're talking molecular imaging. So what do you use to inform on molecular imaging? Well, you need a contrast agent, or you need a probe or a tracer, or whatever it is that we're calling it nowadays. But you can do endogenous versus exogenous. So a lot of my friends in the cancer field, they love luciferase imaging. And it's really been helpful. I mean, when you're trying to validate targets or you're trying to validate biology, what gets turned on where, luciferase imaging is really valuable, and so can be GFP and some of the other fluorescent proteins. And clinically, this has been used for things like adoptive cell transfer, especially in immunotherapy. So putting in the HSTVK, putting those cells into patients, and then coming back with the thymidine analog for PET has been where this endogenous probes have been translated into the clinic. Now, granted, it's very limited application for endogenous probes. I mean, we're not going to genetically engineer things really that go into us. You can't pull out someone's tumor, engineer it, and then put it back in. This just doesn't make sense. But preclinically, this has been really important. And like I said, for cell tracking, this has been pretty important clinically. So that leaves us with exogenous molecular probes. And so to do this, you have to think about a targeting agent. So the form of these agents usually go with a targeting moiety. And that could be something like an engineered protein, an antibody fragment, an antibody. My favorite is peptides, so I'm a little biased there. Aptamers, so DNA, RNA aptamers, all sorts of different things that you can make that are targeting agents. So you have your targeting moiety, small molecules. I forgot about those guys, but small molecules, FDG. I mean, really, right there is one of the prime examples of a small molecule for molecular imaging. And so you have your targeting moiety, usually followed by some kind of linker, and then after that you have your imaging moiety. And most of the time it's pretty modular, so you can cut and paste, and depending on your application. So again, talk to your clinicians, your radiologists, talk to the people who know what they're using, what the application is, in order to develop the best tracer. So from there, think about it, target selection. So we can target almost anything in the body. I mean, there's an antibody to it, right? Or you can make a peptide or small molecule, whatever. This is just the metabolome. So this is from Roche. Oh, I forgot, this is a CME. So I am conflicted, not with Roche, but I have a company that's a startup company for pancreatic cancer imaging. So I'll be talking a little bit about that, but yes, I'm a little conflicted. But anyway, back to this. Here's with the target selection. So how do you choose a target? So here's just the metabolome. How do you look at this and figure this out? Well, the good thing is that we have people that are really excited about big data, and they are working on trying to figure out what to do with all the cDNA microarrays and all the proteomics information and all the metabolome information, and they're putting it into a computer, and hopefully they will give us the recipe for how to choose a target. But I have never been good with computers. I'd like to learn one software program, which is why I still run Prism or something, because I don't like to change. And so this isn't where I am. And so I'm more of a practical kind of person, again, which is why I'm in an engineering department, very pragmatic. And one of the things, so I'm going to go back. Anyway, so how do you choose a target? And now I'm going to put this in the context of an application, because again, you're going to choose your target based on what the application is. So I'm going to get back to how we're going to do this. But what I really want to put forth too is just my humble idea of how we should be making clinically relevant, translatable, commercially viable imaging agents for the molecular imaging and for the clinical community. So my disease specialty is pancreatic cancer. That's where I've done most of my work. And so just for a little background, pancreatic cancer is low on incidence. So it's about the 10th most common cancer. However, it's number 4 in terms of death. And that means that 94% of patients that will be diagnosed with pancreatic cancer will not survive past 5 years. So it's actually been recently labeled as a recalcitrant cancer, and legislation was passed through Congress to give more funding to some of these recalcitrant cancers. So in the past 40 years, the prognosis for patients hasn't changed. And so there's challenges to this. So some of it is detection. The imaging modalities that are used right now for pancreatic cancer are endoscopic ultrasound and also CT. But the way the cancer grows, it's diffuse. It's not so easy to detect. And pancreatic cancer will also metastasize before it reaches 1 centimeter, which makes it challenging for some of the imaging modalities that they're using. So right here is an application tailor-made for molecular imaging. So is there a way that we can get contrast or sufficient contrast that we can start seeing this earlier? Now, because of the incidence, so again, this is thinking about applications, because of the incidence of the cancer being low, it's not like a mammogram, it's not like colonoscopy, where I actually heard that 1 in 3 patients, or 1 in 3 people that get a colonoscopy will actually have polyps, and those will be removed, and theoretically you're preventing colon cancer. This is not particularly the case for pancreatic cancer. Because the incidence is so low, we just cannot afford to screen everybody. And with the high false positive rates potentially, then you would be doing a lot of things that would not be necessary clinically. So we have to start thinking about, you can't just use an imaging test for this. So you'd use things, you'd start to put people into bins or high risk groups. So family syndromes, patients with new onset diabetes, so people that are a little bit older and that have a normal BMI, but then all of a sudden get diabetes, they would be worked up for potentially having pancreatic cancer. Cystic lesion. So there's over 100,000 people in the U.S. that have a cystic lesion in their pancreas. Most of them are benign, but the few that do have a cancer associated with them or will become cancerous, then they have a very bad prognosis. And so being able to tell which lesions are benign versus which lesions are malignant is a really important value add to this. And then finally, a positive serum test. So right now, people in the field of pancreatic cancer are looking for a serum test. So in this case, you'd have something like a CA19-9 or things that people are coming up with. Those would be positive, the serum test, and then you'd go do the imaging. Because we know in serum tests, even though it will tell you that there potentially is the presence of a cancer, it doesn't tell you how much, it doesn't tell you where, and the rates of that marker appearing in the serum does not tell you or correlate anything with patient prognosis. So even if there's a good serum test, there's always room for molecular imaging. So this is work from Jason Lewis at Memorial Sloan Kettering, and so CA19-9 is the only serum test available right now, and that's been mixed reviews. Basically, I mean, it's not really that informative. It has low specificity and low sensitivity. But what Dr. Lewis's group, what Jason's group has done, is they have a human antibody, so not a humanized antibody, but a human antibody against CA19-9, and they have started to image it preclinically. And so here's the PET scan, there's the tumor on the left. So not only have they done this in xenografts on the flank, they've also done this orthotopically, and in pancreatic cancer this is very important because of high desmoplastic stroma, and then the other hallmarks that were talked about today with the leakiness of the vasculature, resulting in high interstitial pressure, so on and so forth. And so he's actually done this, or his group has done this in an orthotopic model. And if I know Memorial Sloan Kettering, I have to admit that I'm incredibly jealous of their infrastructure, their IND writers and their protocol developers and their radiopharmacist. Serge is amazing. But they will be probably taking this into the clinic in the near future. So I'm really excited about that. And then from there, so CA19-9 was already reported. It was already in the literature. So how, again, coming back to the theme of how do we choose targets? Well, you do a literature search. And if there's a good target out there and someone has done all the work for you and done all of the IHC studies and all those things, that's a good target. Now you build an imaging agent to it. But what my group has been doing is we've been trying to find targets that aren't in the literature. And so what we've done is we've modified a phage display protocol to do functional proteomics. So not only when we do phage display do we get the peptide that we can then use as an affinity ligand or a targeting moiety for whatever modality that we're interested in or targeted drug therapy, we also have developed methods to go back and identify the binding partners of each of those peptides. And in that way, we're querying the cell surface and identifying new targets of the disease and new biologies. So I won't go through too much of the phage display, but we use the internalizing protocol where we have the cell, we let the phage bind to the target cell, in this case pancreatic cancer. We let it internalize, wash away everything that doesn't bind, elute the cell surface bound, and then lyse the cells to get back all of the internalized. From there, we do a subtraction against normal cells. In this case, these are cells from the normal pancreas. Then do ELISAs to validate. We validate on mouse and human. In my lab, we require that it cross-reacts both mouse and human. So not only does the peptide cross-react, but the epitope has to be present in a mouse cancer and a human cancer. And we do that because most of the models that we need to use are mouse models. I mean, we have to do this in the mouse. And so we are losing things, absolutely. But it's really important to be able to take this. And again, I really want to be translational functional. And so I want to be able to take this to the clinic. So that's why we require that. From here is the functional proteomics part. So we have done a lot of chemistry. My undergraduate degree is in chemistry, so I'm one of the people that loves organic chemistry, so I don't have to say anything about that with this group because you guys, most of you are on board with organic chemistry. But we use the phage as an affinity ligand or a handle. So we use a photoactivatable cross-linker, and we put that onto the phage through the free amines on the phage coproteins. So there's 2,700 P8 coproteins in a phage. We use it at a ratio of about 1,000. So the idea is we're putting about 1,000 of these photoactivatable cross-linkers on there. We also put some biotin on there so we can use it as a handle to fish out. These are I don't want to say nondescript, I'm blanking on the word. But anyway, these we do not control, they're not site-directed. We are working on methods in our lab to start doing site-directed labeling so we can put a handle on it and not just have statistics dictate where it's going to go. But we allow the phage to bind. We do it at 4 degrees because we do not want it to internalize here. We then cross-link it, activate the cross-link with a UVA, UVA, I am from UVA, but UV lamp that activates the cross-link. We then go ahead, lyse the cells, and then using just a magnetic bead that's streptavidin coated, we can pull that out, wash away everything that didn't bind to the bead or in the phage, break the cross-linker with DTT because it's just a sulfhydryl cross-linking, and then run that on a gel. So we save half for further analysis. The other half we run on a gel. We do some kind of Kamasi stain, cut out unique bands, send that for mass spec sequencing. So in-gel terpsic digest, mass spec sequencing. It comes back and we get a list of the proteins that it is. So from there we do now the validation. So we get some proteins that it could be. And we run the other half of the lysate or a portion of that lysate, and we do a Western blot if there's antibodies available. And also, we either purchase the purified protein and absorb that and do an ELISA assay to functionally validate that it was the binding partner, or, I mean, in this day of molecular biology, we just clone it and make it and purify it, and again, we do a purified protein ELISA. So, and from there, from that original screen, we were able to identify at least 5 new things on the cell surface of pancreatic cancer. Pyruvate kinase M2, which has then since been shown to be on the cell surface, even though it's not a cell surface protein, it's actually cytoplasmic. It's been shown to be on the cell surface, not just by us, but another group. Annexin A2, so Liz Jaffe's group in Hopkins actually showed that this protein is on the cell surface through a phosphorylation event. And also, it is present in the immune response for when they're doing immunotherapy, Vimentin. But then this protein, Plectin. So Plectin is the protein I'll be talking about. It's a 500 kilodalton protein, and actually it's highly upregulated in skin, in keratinocytes, highly upregulated. And when it is mutated, patients have epidermolysis bullosa. So that's a skin blistering disease. It's responsible for maintaining integrity of the junctions, the hemidesmosomes in the skin. So what is it doing on the cell surface? Well, we found out that it's in an exosome-mediated mechanism. So Plectin actually controls exosome formation. Without Plectin, the cells do not make efficient exosomes. And it changes the proteomic profile of those exosomes. So with Plectin there, you bring a whole bunch of ribosomal proteins and other protein synthesis proteins, plus some MMPs. Without Plectin, that's just not the case. But it also acts as a scaffold for cellular signaling, so there's an outside function, there's an inside function. With CXCR4, RAC1 PKCs, PI3 kinase AKT, MAC ERK1 2, so a lot of the cell signaling pathways in cancer, Plectin is involved in scaffolding for them. So we did all of the validation. So we found this target. We went back to patient samples, so over 100 patient samples, and started looking where it was expressed. So it's not in normal pancreas. It's not in chronic pancreatitis. Chronic pancreatitis is a risk factor for pancreatic cancer. So again, choosing the negative tissues to look at is important. Pannins 1 and 2, so pannins are pancreatic intraepithelial neoplasms. They are precursor lesions. Pannins 1 and 2 are pretty common. About 30% of people on autopsy will have a pannin 1 or 2. The surgeons would not remove those. They just wouldn't. But a pannin 3 now becomes an advanced precursor lesion or a ductal carcinoma in situ. And so a surgeon wants to know those lesions are present, and then they would remove them, thus like a colon polyp preventing pancreatic cancer. So Plectin starts to be expressed in pannins 3, right here on the cell surface. In PDAC, it's cytoplasmic and in the cell surface. It's in the liver, lymph node, and peritoneal metastasis, but it's not in the background tissue. So it's not in the liver, the lymph node, or the peritoneum. And then this is a xenografted PDAC. And so this is a chunk of patient tumor that's been implanted surgically into the animal, so an orthotopic model. And we just wanted to check and make sure that Plectin was still there after we did the model. We also looked at cystic lesions. So I told you before that cystic lesions and predicting which will become cancer or which have cancer associated is really important. So we looked at that, and it turns out that Plectin is highly upregulated in the cystic lesions that have the presence of cancer. So that's really exciting. We looked at other cancers, head and neck squamous cell carcinoma, esophageal cancer. These have since been validated, and in fact, Plectin is a prognostic indicator for head and neck squamous cell carcinoma. So it's really exciting validation of the results. So from there, we got excited. It could be a biomarker, or it is a biomarker. And so now, can we make the imaging agent? Well, from the phage screen, we already had the peptide. And in this case, we decided to go multivalent because the monomer isn't really high affinity. But when we make it multivalent it has a higher affinity through avidity effects. So we tetramerized it. Put some peg chains here, and then used it for an indium-111 for SPECT imaging. From there, these are the orthotopic animal models. So here's the tumor in the pancreas. There's the kidneys. It's a smaller molecule, and so it gets excreted through the kidneys. And then here's peritoneal metastases that we're imaging. Here's just different animal models. So again, this is a human tumor in there. This is a mouse tumor, so this is a syngeneic. Here, and then that's the null animal, so sham operated animal. We also did liver metastases because one of the major sites of metastases and an important application for pancreatic cancer is determining whether patients are surgically resectable. If they have liver metastases, they are not surgically resectable, so they wouldn't go through this enormous surgery procedure. And the procedure is to take out the majority of the pancreas and replumb the stomach and in the duodenum, and they take out a third of the duodenum. And so it's a major surgery, and if they're not going to benefit from it, then there's no reason to do the surgery. And so we used a mouse model where we injected tumor cells into the splenic capsule. They migrate up the hepatic portal vein and then deposit as metastases inside the liver. And so we were able to image the liver metastases. So that agent, actually, we have FDA approval. It's been through the IRB, and so we are currently enrolling patients, and so we are moving it. And it's through an exploratory IND mechanism. The FDA was very nice about it, and so I would encourage you to translate as quick as possible into the human, because I've done all the mouse models I can, and people say, well, you know, what about this? And well, I don't know. I have to get into the human, and so that's what we're doing. We are moving into the human. So we have the scouring the literature approach for target identification. We have the phage display, functional proteomics that I've talked about. But then Julie Sutcliffe from UC Davis, who's just a fantastic researcher and one of my good friends, she used the one bead, one compound approach. And how she did this differently, which I thought was just absolutely fantastic, this is in a PNAS paper, is she did this for imaging. It wasn't just imaging as a byproduct. She did all the one bead, one compound, but then she did high throughput PET imaging and developed a platform to do the high throughput F18 labeling, so then she can do all of these animals. So they screened 43 different compounds in these animals to come up with their compound. So the quicker you can start doing it for imaging, the better off it's going to be. And when you start setting your metrics or your parameters for success, thinking about the imaging modality and the application and what is going to be a successful imaging agent becomes really important. So again, getting it in vivo as quick as possible and not just relying on the in vitro information is really important. So from all that screening, she came up with a peptide, and then from there was successful in being able to image pancreatic cancer by PET in these animals. And so I'm hoping to see her stuff in the clinic pretty soon too. So really great year, hopefully, for pancreatic cancer and pancreatic cancer imaging. And hopefully one of these tracers will really work and allow clinicians and patients for better patient prognosis. So that's just the tumor. What of the microenvironment? So this is, again, with my Plectin agent. Here's the tumors. Here's just the long circulating fluorescent dextrans. So now we can start to look at the tumor and its microenvironment. This is in a transgenic mouse model. So unlike the orthotopics where you only have one tumor, that's one of the caveats of these transgenic mouse models. You get hundreds of tumors all through here, but we're able to see the individual cells streaming through the stroma. And this is why this streaming through the stroma is exactly why it's difficult to image pancreatic cancer with CT or with ultrasound, because they'll start to metastasize and do this relatively quickly. But from here, one of my friends, Terry Bretnell at University of Washington in Seattle, she did a classic proteomics approach where they ground up tumors from a family. They ground up tumors versus normal, and they looked for changes in the proteome. She came out with this protein Thi1, which is a vessel marker. So she's looking at markers on the cell surface of the tumor vessels, even easier. From there, she validated, again, lots of validation, so of these different things. So I don't know if you guys were at the chairman's talks, but again, talking about the importance of pathology and validation in molecular imaging, everything is so multidisciplinary. But she validated it again in chronic pancreatitis. It's not there. It's in the blood vessels of pancreatic cancer. And then what they did is they put it on microbubbles. So microbubbles are perfect for imaging vessels. You can see down in some systems the single bubble resolution. So they put the Thi1 antibody, so not a peptide, but Thi1 antibody on the bubbles, and we're able to see in transgenic mouse models those pan in. So those precursor lesions that aren't cancer. So here's the control microbubble, here's the Thi1 targeted, here's the pancreas for the chronic pancreatitis, here's the normal pancreas, and then this paper has been published in gastroenterology, and they have all of the lovely work done with the pan-ins and really being able to see at a very low level this. So ultrasound is a great modality in terms of cost, and really being able to see this in real time too. The targeted microbubbles haven't really made inroads into the clinic, and I'm not sure why, and I'm sure people will know. So I'd really love to hear about that, is why people think that the targeted microbubbles haven't really made it into the clinic. And whatever that is, if it's an FDA barrier or whatever kind of barrier it is, I think that being able to do these microbubble experiments could really be helpful, and especially in this case. So with that, I'd like to close and thank the members of my lab, Stephanie Thomas, who's doing all of the clinical things, Mark Seaman, Sue, and then also Julie Sutcliffe and Jason Lewis for letting me have their slides. So with that, I'll take any questions, and thank you.